Think forward. Think Research Channel.
So this is really an incredibly new area in terms of dealing with cancer. I'm an oncologist here at Stanford as well as a geneticist, so see patients with cancer. And over just the last 10 years, really, our understanding of genetics and of the human genome has gotten to the point where clinically we can actually use this information in some situations, particularly to try to identify the underlying genetic problems in families that have a very high predisposition to cancer or a very high incidence of cancer. So one of the important points to start out with is at this point, this is still a very young field, things are changing very fast, and like in most things in medicine and science, we understand the most extreme examples of things first. So we have a lot of information now about families that have a very striking predisposition to cancer with multiple relatives and many generations, and I'm going to show you examples of that tonight. But that, of course, doesn't account for the large proportion of cancers. Maybe 20% of cancers fall into that category. So a lot of what I'm talking about today is going to be our clinic and for this purpose. And so a lot of it is who is this really appropriate for? What kind of families should you proceed with genetic testing? And if you do, what are the things that you can actually do about it? Because it's not yet for everybody. But I do think over the next decade, a lot of these principles will extend to the much more common cancers, and we can assess risk in a better way. This whole field really just started in the last 10 years, and I've been fortunate to start a clinic at Stanford, our genetics clinic, about 6 years ago. So the primary types of families and patients that we see in the clinic are ones with the following types of cancers that seem to run in their families. And as you'll notice, most of these are the fairly common cancers, but as I'll show you, that their presentation of these families is uncommon. So we know a lot now about families that have a predisposition to breast and ovarian cancer, and I'll tell you about examples of this tonight. We know a lot about families that have a predisposition to colon cancer, and I'll tell you about this tonight, and these are just different syndromes that we see. There are also lots of other syndromes or hereditary cancer families that we're understanding and actually finding some of the genes that we can test. These are much more rare, and I'm not going to use those as examples tonight. And there's certainly lots of other cancers that run in families that we really don't have much of an idea what's causing them yet, but I'm sure that we will over the next few years. So one of the first things I need to do is just go back and take you all back to high school biology and high school genetics and make you think about these things again, which you may not want to, and talk about what is a cancer and how does genetics play into that, and then some of those basic genetic principles like the yellow peas and the green peas and how we actually use those now. It applies to humans just as much as peas, not that Mendel knew. So what's a cancer in terms of genetic causes? Well, the simplest definition is that it's a cell that has lost its control, a cell in our body has lost its control over its division, over its growth, and has grown out of control and divides many, many times and creates a mass or a tumor. But why does that happen? Well, our best understanding for common solid tumors, particularly like breast and colon cancer, is that in the normal cells in our bodies, some event happens to the DNA in that cell. It undergoes a mutation in a gene. And this predisposes it to start losing this growth control. But that in and of itself probably isn't the whole story with cancer. It probably takes many, many mutations and many, many different genes till you actually get to a place where you have an invasive cancer. So we think that these very early events tend to be ones that somehow cause instability in the DNA or predisposition to acquiring all these mutations. And what I'll show you is these familial syndromes tend to inherit problems in just those kinds of genes. So they already have this inherent instability in their genomes in all their cells, leading to these many other mutations. So this just summarizes that the cancer clearly is a genetic disease. We know that mutations in genes cause it, whether it's in families or whether it's just by chance. Most of them have multiple genetic changes by the time it's a tumor. And as I said, the underlying defect tends to be this instability in maintaining their DNA. So most inherited cancer syndromes then are due to alterations in these genes, and that's how we've learned about the very early stages of cancer genetically. Well, so the most important thing then is first to decide just what kind of cancers are really hereditary or seem to have a familial basis, and which just happen by chance, maybe due to environmental factors, certainly genetic factors, but maybe not ones that are inherited within a family. And so we make these definitions about sporadic cancers, which is the majority of cancers, probably 3/4 of cancers, of the common cancers, lung cancer, breast cancer, colon cancer. And the families in which those occur don't seem to have any higher predisposition to it than in the general population. But probably about 1/3 of cancers occur in families that seem to have more cases than you would expect. And this could be breast cancer, colon cancer as well. But what we define as hereditary cancers are these days about 10 to 20% of cancers that have a clear pattern of inheritance, and I'll show you that in a minute, that we call dominant inheritance, that occur at a particularly early age in families, and individuals in these families might get multiple cancers, so it's a fairly dramatic occurrence. And it's in these families that we can actually identify what we call germline mutations. That means a mutation in that gene, not just in the cancer cell, but actually in every cell in the body that can be passed on to the next generation. Well, these are some of the examples of the red flags or the cardinal features of a family that would fit into one of these hereditary cancer syndromes. And that's when you see a family in which there's individuals with cancer at a very, very early age compared to the normal age span for those kinds of cancers, or that they have multiple different cancers in the same person over their lifetime, or multiple cancers in many individuals in that family. Some of these syndromes carry other physical or pathologic features that help us determine these. And what's important is that in some of these situations, the cancer that occurs in these familial situations actually has different outcomes, responds differently to treatments than the same cancer that occurs by chance. And so it actually helps us to think about treatment and prognosis as well. And then what I'm going to tell you about is the pattern of inheritance in these families and how we try to spot the families in which this is relevant. So before we get to that, one more drag back to high school genetics, just to make sure we're all on the same page. So when we're talking about mutations and genetics, what we're really talking about is changes in the primary DNA. So DNA is just a strand of chemicals that's billions of letters long. It's just like a book with words and letters. And the sequence of all these letters make up a code for proteins that encode a function. And a gene will encode a protein. And that can do lots of things. It can metabolize sugar, or it can control cell growth, which will be important in cancer. Well, these are all wrapped up, and that's when you hear the words chromosome and nucleus, and they're all stuffed into one cell. And there's about 23 or 25,000 different genes in our genome, and that's the best estimate these days. Well, when a mutation occurs, what that means is just that potentially just one of these letters out of the 3 billion was altered for some reason. Maybe because it got damaged from some environmental insult. Maybe it was just a mistake when the cell was copying its DNA. Those things happen all the time. And if it happens at the right place, and if it happens in an important gene, it might cause an alteration in the protein that's made so that it doesn't work as well. So if this mutation occurs in the germline, and thus it can be inherited through the sperm and eggs in the next generation, this will then get passed on and copied faithfully in a child from that, and spread into and be in every cell in that person's body. The other important part of genetics is that we have 2 copies of every gene. So we call these chromosomes. And we have 22 chromosomes in each of our cells, and then a set of sex chromosomes, X or Y chromosomes. And we inherit one of those from mom and one of those from dad. They theoretically are identical, but because they come from different individuals, there might be small changes. And so if one parent potentially had a mutation in a gene and the other didn't, there would be a 50-50 chance that that would get passed on to one of the progeny. And so that explains the hereditary basis of these cancer syndromes. So in the sporadic situation, non-hereditary situation, we think that both copies of that gene have to somehow get mutated or deleted, because if just one is deleted, the other one's probably still working fine. So it means that there has to be two events that happen at the very same gene out of these 23,000 genes in the same cell, an incredibly unlikely occurrence, although obviously it happens because cancers are happening. In the familial situation, though, the hereditary situation, a mutation in one of those genes has been inherited. It's already in every cell in the body. So all it takes is one more hit or change in maybe any of those cells to confer this disposition to proceeding towards cancer. And so in this situation, the person is what we call one hit closer, or has already acquired one hit. That's why these cancers occur at a younger age. It takes less time for these things to happen, and maybe happen in multiple places in the same person, because all the tissues are potentially at risk. And so the last bit of this then is why we call this a dominant condition. And that's because there's a 50-50 chance of passing on this change to any child. So if here dad has a mutation in one copy of those two genes, mom is normal in both of those two genes, each child has a 50-50 chance of inheriting one or the other of those genes from each parent. And so it's just going to be a flip of the coin whether they got the faulty gene from dad or they got the normal gene from dad. And so when we look at these families, we tend to see patterns where about half of the children in each generation may have the effect. So this is true for any genetic syndrome, not just cancers. And in cancer, what's getting passed on then is the risk of getting cancer. So it's not like a child in each of these situations has cancer, but they have the increased risk. So that's what the 50-50 chance is. And so here's just an example of the sort of pedigrees or family histories that we get in the clinic and try to decide what the risk is. And so when we look at these pedigrees, just like a family tree, the squares are men and the circles are women, and these are 3 different generations. And in this case, the orange is denoting that there's a trait in that person, in this case, a cancer developed. And you can see that the grandfather in this family tree developed a cancer, and of their children, well, 1 out of 4 developed a cancer. But then in the next generation, you can see 3 of these 5 did. So on average, you're getting towards this 50-50 split. The flags about hereditary cancer syndromes is there's vertical transmission, or there's passing on multiple generations of these problems. And sometimes it has to get passed through somebody. We know that this trait had to go through this person to get passed on here, but they don't have the disease. And that's because, as you'll see in these families, we never know, these cancers might develop at a very early age, but they don't necessarily. It might be much longer. And what we don't understand is this variability. Why is one person in the family who's affected with a genetic change have a different outcome than others? So the pedigrees aren't perfect, and we sometimes have to account for these skipped generations. Given all that, how do we use this information? How do we really decide who we should test, and how do we do this testing for genetic susceptibility to cancer? What are the actual risks of cancer associated with some of the genetic changes that we've been working on? And if you find this, what can actually be done? The only point, of course, in proceeding with this, we hope, is that we can do something to either help the person who's developed cancer or prevent cancer in their relatives. When should you actually think about genetic testing? Well, it should be if the patient and their family has a reasonable likelihood of carrying one of these cancer susceptibility genes. And that's when we use all the criteria I was telling you about, that there's multiple generations, that they're young and have multiple primaries. It doesn't make sense to pursue this in a family that doesn't have a very high risk, because you sometimes can go off on a wild goose chase and not really get any useful information. Of course, we have to have a genetic test that's available and can be interpreted, and that's changed dramatically over the past 5 to 10 years. We really didn't have these 10 years ago, and we'll have a lot more of them 10 years from now. That the results of this will somehow influence our management of the patient or of the family or of both. And of course, that the patient wants the information. This is certainly not something that should be done in an anonymous way or as a population screen. I think perhaps the most important part of this whole process is, and unique in terms of my medical experience, is we work with genetic counselors in this situation. So specifically trained to gather these family history information to help interpret the risk, and there's sophisticated statistical tools now to calculate risk based on patterns in families, and then to educate and inform the families themselves so that they can make an informed decision about proceeding with this, what the risks and the benefits are. And then teaching them about the basics of genetics. Exactly the sort of talk I'm giving you today is what we do in the clinic, what the genetic counselors do in the clinic every day, and spend a lot of time with these families. So the process we use in starting this is really, the counselors are assessing the risk in the medical history, educating families in terms of the risks and benefits, and understanding that these aren't perfect tests yet. We sometimes get a negative test, and that doesn't necessarily mean that there's no risk in the family. It just might mean that we don't know what to look for yet. We haven't discovered all these genes. And then, of course, to talk about some of the options should a family pursue testing and find out they carry one of these alterations. So the genetic counselors then, of course, take a family history, and it's very important, as I was showing you, that we get 3 generations, that we try to find all the relatives and really find out what their diagnoses were. A lot of information that a lot of families don't have. So this can sometimes take quite a lot of time to track down relatives, maybe from other countries, and often a long time ago to get the information. And then discuss the benefits and the potential risks or limitations of testing. Well, we think the benefits are that we can improve the management of cancer and in preventing cancer in these families, at least in certain situations that I'll tell you about tonight. Certainly, in testing relatives, as I told you, there would be a 50-50 chance in many of them. So finding out that one didn't inherit a mutation that's running in their family would obviously could provide relief from worrying about that risk. And finding out one did might inspire certain lifestyle changes to try to prevent cancer, or pursuing various screening tests to try to pick things up early if they should happen. But there are worries about the limitations, and that includes dealing with the results of tests, big issues in terms of loss of privacy, potential insurance discrimination or job discrimination. I'll touch on that in a minute. And then still, many of the interventions or our ability to try to screen and prevent cancer in these situations aren't really yet proven. We don't have as good a tools for this as we'd like. Now, many people are very concerned about insurance, as you know, with genetic testing, that by having a test like this done that might get into their medical record or be identified by an insurance company would put them at risk for losing insurance, but also in terms of does insurance actually pay for these tests? They're very expensive. So this is another reason we do a lot of work calculating risk, because insurance companies will rarely pay for tests unless there's a substantial risk or a reasonable risk that one exists. And as I'll show you, we're going to talk about the breast cancer and colon cancer, and we use, if we calculate that there's a risk of about 10% or more in that family of having mutation, that's usually paid for and supported by most insurance companies. Medicare uses fairly similar criteria these days. Medi-Cal is we almost never can get coverage. So there isn't universal coverage for these tests these days, and they can be thousands of dollars. So it's a big issue for many of our families. In terms of discrimination, there are fairly good laws, but they need to be better. There are California laws that prohibit genetic discrimination by employers. There are some federal laws, particularly in this new HIPAA Act that you may have heard of, the Health Insurance Portability Act, that legally prohibits insurance genetic discrimination, but that's only for group insurance. So that, of course, isn't applicable to everybody. At a local level, we are incredibly compulsive about not letting this information go out in some general way. Our notes, the results of the tests, all of our recommendations don't go into the medical record at Stanford. We maintain a shadow account that's held behind firewalls. And we would only reveal that information to other doctors or other individuals that the patient has let us consented that they want that to be sent to them. So we do as much as we can to maintain privacy. And the truth is, for all the worry about this, there has been very little real documented evidence of discrimination. That doesn't mean it's not potentially going on, but it seems to be less than we worry about given the situation. Okay, so I want to talk about 2 examples of of hereditary cancer syndromes tonight. They're the most common ones we see. They constitute probably 2/3 of the families that we see in the clinic. And that's our hereditary colon cancer families and hereditary breast cancer families. But really all these principles apply to a lot of the more rare things as well. So in talking about colon cancer, just some basic information, extremely common in this country and in the world. It's the second most common cause of cancer deaths in the United States. The lifetime risk for an average person, not from these particularly high-risk families, in their lifetime is about 5 to 6%. So over the course of an entire lifetime, about 1 in 20 individuals will develop colon cancer. So it's a fairly common cancer compared to others. The vast majority of them occur in individuals that are over 50 years old. So when we see individuals who are much less than 50, it's a real signal for us that there might be something going on in the family. And about 1/3 of these cases have some family predisposition. I'll show you. Maybe one relative that has colon cancer or a distant relative. And about 10% will have the very dramatic families that we'll talk about in terms of who we can do genetic testing. So as I said, if you look at all cases of colon cancer, about 2/3 of them seem to be sporadic. They happen by chance. We're not really sure why they happen. And the families don't seem to be different than the normal population. But about 1/3 of them have some family basis. There's more cases than you'd expect. So they must have some genetic predisposition. But at this point, really only about 10% do we really understand and can do genetic testing. So we have a long way to go in terms of figuring out what's all going in here, because we really don't have good tests for assessing risk in those family members. But remember, if there's almost nearly 200,000 cases of colon cancer a year, this 10% is still a substantial number in this country, and that's why we still have a lot of families coming in. Well, there's some very specific aspects of this syndrome that differ it from the sort of sporadic colon cancers. So the things that make us think about this is families in which an individual or several individuals get colon cancer at a particularly young age, under 45 is a big signal. They tend to be in the right side of the colon much more often than sporadic colon cancer does. And there are other associated cancers that fit into this family syndrome, not as common as colon cancer, and I'll show you the data in a minute, but endometrial cancers, ovarian cancers, a number of other GI cancers. If we see those scattered in a family, that really makes us think of this syndrome. And I'm sorry, I forgot to tell you, the syndromes was named by Henry Lynch a number of years ago, so in medicine that's how all these syndromes get named, but we really call it hereditary non-polyposis colon cancer. And this is a bit of a misnomer because it doesn't mean you don't get polyps, but you don't get hundreds of polyps, which is another very rare disorder. So the formal criteria that people have used for years for who to do genetic testing for this are really quite stringent. So, we don't need to go through this in detail, but it's families in which there's 3 or more relatives who have colon cancer in 2 or more generations, that one of those individuals is less than 50. So this is a pretty dramatic family, and we don't know too many families like this. But of these, if one goes ahead and does genetic testing, and as I'll tell you in a minute, the genes that cause this are called mismatched DNA repair genes. If we go in and look at these genes, about 2/3 of these families will find an abnormality. So using these very strict criteria, we're pretty good at identifying which families. These days, because the cost of testing has gone down, because the ease of testing is much different, and because we have some ancillary tests to help us pick up these families, we have much looser criteria. In addition to all these sorts of things, really anybody who has colon or endometrial cancer at a young age, less than 45, we would now consider looking into this. The percentage of these that end up having this syndrome is less, but it's still almost 1/3 of these folks. So what are the genetics? Well, it has this pattern I told you about a 50-50 chance in each generation of passing on. There's a high degree of getting colon cancer. In about 80% of individuals who inherit this will, in a full lifetime, in 70 years, will develop colon cancer. And here, there's a series of genes that cause it. MLH1 and MSH2 are just the names of these repair genes. And these are the things that we can test for by sending simply a blood test from these individuals. And the clinical laboratories can now sequence the DNA in those genes, read those thousands of letters that make up that gene, and determine if any one of them is different from the expected one. So just to put this into context, In families that we find, or individuals in whom we find a mutation in one of these genes, they have a very high risk of getting colon cancer, 70 to 80% in their lifetime. In the general population, I told you the risk of getting colon cancer is on the order of 5 to 6%. But of course, there's a spectrum in between. Not these dramatic families, and these are all the families that was the slice of the pie that we just said there's a familial basis. We don't really understand it, but they definitely have increased risk. So if you have several relatives that have colon cancer, you may not have this syndrome, but your risk of getting colon cancer is a little bit elevated. And those are the sort of numbers we use to help guide people in terms of colonoscopies, how often they should get them, at what age they might want to get them. But I'll tell you mostly about this dramatic syndrome and what we are trying to do for this situation. So this just shows these families. So it's looking at the incidence of getting colon cancer over time, over years, and it just goes up steadily, starting at a really early age, 30 and 40 years, up to 80%. But there's a higher risk of a lot of other cancers in these families too, particularly endometrial cancers in women. There's a nearly 50% chance. So we need to try to deal with screening and preventing all these different cancers in these families. So what's the process? We try to start with doing genetic testing in an affected family member, meaning we want to start with somebody who actually has colon cancer. A really common situation is, of course, we see a family member whose parent or grandparent has cancer, and there's a number of cancers in the family. They themselves are well. Doing genetic and come in and want to know, did I inherit this risk? Should I be worried about this? Well, it's hard to do testing on that person without finding out if the family has a mutation. And so optimally, we like to do the testing in somebody who actually has the condition. And then we can, if we find it, we can test the various relatives. If that person tests positive, then you can offer testing to the other at-risk family members. That initially would be any first-degree relative, and then go out in the family based on that. Remember, 50% of them won't have this mutation. So if they have a negative result, here we also may not be able to identify a mutation, and that doesn't really help. That means that the other relatives do have to assume they're at some increased risk. Well, can we do anything about this if we find this mutation? Well, this is where a lot of our clinical work comes in. We certainly are able to prevent colon cancer in families in whom we know they have this genetic risk by performing colonoscopies and starting at a really early age, starting at 20 or 25, because you saw on that curve, 20 and 25-year-olds were starting to get cancer in these families. So by starting at that early age and doing colonoscopies every 1 to 2 years, we have decreased the risk of getting cancers and certainly increased survival, and I'll show you that information on the next slide. What about these other cancers? Well, we've screened for those as well, but we're not as sure how efficient the screening is. For endometrial cancer, you can do tests like ultrasounds and examinations, and for some of these other cancers, there's other endoscopies you can do. And so we recommend these tests, and we do them. It's been difficult to prove if we're making as much of a difference as we are with colonoscopies. And this just shows the incidence of or the survival following in these families, whether they have yearly or bi-yearly colonoscopies compared to families, HMPCC families that do not. And you can see that we've made a big difference in picking up at least the majority of the cancers that occur there. Well, it'd be much more optimal if we had a pill that would just prevent this, right? And so when we talk about chemoprevention, what that means is, can we identify some sort of a drug or therapy that would prevent the cancer from ever occurring? And there's a lot of work going on this. There was a big setback, as you know, from this. So it was in this exact population where this whole story about the COX-2 inhibitors occurred. There was information from big studies that the drugs like Celebrex or Vioxx or Motrin or aspirin, a lot of these kind of non-steroidal drugs, may decrease the incidence of getting polyps and may actually protect against getting colon cancer. So many large studies over the past few years got started with using these drugs as preventative agents, and they started in these very high-risk families, just like this, because we had the best chance of showing an effect. In fact, they do seem to slow down polyp formations, and they probably are pretty effective. Unfortunately, as you all know, it turned out when they did these huge studies with thousands and thousands of patients, they increased the risk of various cardiac problems, and that led to everything this year, and these drugs getting withdrawn from the market. And so it's an unfortunate problem. We have agents that probably do help to some extent, but they have a side effect that is hard to deal with. And that's the problem with these prevention strategies. You're putting lots and lots of healthy people onto a drug that may have side effects, and even if the side effects are low, if you have that many people, it's going to occur fairly often. So this is the tough thing in balancing some of these prevention trials. So we're kind of back to square one in this case. Well, so what are the risks and benefits then of doing testing in these colon cancer families? Well, the benefits are that you might identify who is a carrier and thus be able to provide better screening. The limitations are that the tests aren't perfect and we don't always pick up everybody. But that's changing a lot, and we're at about a 90% level these days. You can identify who didn't inherit that mutation, who's not a carrier and doesn't need to worry about this very, very high risk. That doesn't mean they don't have any risk of getting cancer. They have the same risk as the general population, so that they go back to the normal screening rules. And that we've shown that at least colonoscopy is very helpful in preventing cancer in these families. We don't quite understand about the other cancers, but we're certainly working on that as well. All right, well, I want to turn to the second example, which is even more common, and that's hereditary breast cancer. So there's lots and lots of factors that affect risk of breast cancer. Only one small part of it is the hereditary syndromes I'm going to tell you about tonight, though they're one of the most dramatic ones. But as you know, age, just family history in general, various menopausal and hormonal histories of families, dietary factors, environmental factors, probably all impact on the risk of breast cancer. So it's a very complicated field to make good predictions. Again, if you look at the whole pie of all the cases of breast cancer, nearly 200,000 a year that are occurring, once again, about a third of them have some family predisposition. And right now, again, about 5 to 10% of that pie is a hereditary cause, and we understand the genes that cause these. And the genes that cause these are called BRCA1 and BRCA2, and I'll talk about those more in a minute. The features of a family that would really make us think about having a mutation in these BRCA genes, or breast cancer genes, are, again, multiple cases of breast cancer at a particularly young age in a woman, and ovarian cancer in the family. So it turns out this syndrome actually puts people at risk for both breast and ovarian cancer. So if we see a family that has both those things, that's a very high indicator of a problem in this way. If we see it in the same woman, that's an extremely high risk factor. Or if we see multiple breast cancers that occurred in the same woman, even at different points in their life, that's a high risk factor. In this case, heritage actually plays a part in that certain populations, and we particularly know this about Ashkenazi Jewish populations, but there's others from Iceland, populations that at some point in history had sort of a narrowing so that they are often more related to each other than to larger populations. They just by chance have certain genetic characteristics that got passed on in those populations. For whatever reason, in the Ashkenazi population, the BRCA mutations occur at a higher incidence than in other populations, about tenfold. So any breast cancer at a young age in the setting of being Ashkenazi Jewish makes us more suspect as well. Male breast cancer is a very rare thing. You probably know very, very few people, if any, that have ever had that, but it's very predictive of these high-risk families. So when we see that, we think of these families as well. And this is one of these pedigrees that I won't go through again, but this just shows you how dramatic these families can be. So here, the orange is showing women who've developed breast cancer or ovarian cancer at fairly young ages, and this was our patient that came in who had breast cancer at 36. So 3 generations, multiple individuals, breast and ovarian cancer, a young age. When you add that all together, the chance of us finding a BRCA1 or 2 mutation in that person is pretty high. It's probably 2/3 or better. And as I said, these are the 2 genes that we understand that confer the greatest risk for this familial hereditary breast cancer. There are other genes that we think of, but they're very rare at this point. And there's other genes that have not been discovered, so we don't have the whole story figured out, and so there's work to do in this field. How do you test for this? Well, it's the same thing as in the colon cancer situation. You get a blood sample from which DNA can be extracted, and the lab sequence that entire gene. Turns out these are huge genes. I'll show you in the next slide. And at this point, we can identify about 90% of the time a mutation. So it's not an absolutely perfect test, but it's getting pretty close. It's an expensive test. It's cost $3,000 to sequence those two genes. And so this is still a big factor if this isn't covered by insurance or for some other reasons. And so not everybody has access to this testing, in our society or in the world, and it's a big problem trying to achieve this. Well, so these are huge genes, as I said. There are thousands of base pairs, 11,000 letters in this one. So to sequence this means the lab has to read every single one of these at an incredibly high resolution. And it gets to the point where it's going to and finds a single letter that's been changed. And that's all it takes to knock out the function of this gene sometimes. So it really has been a technical feat to get to the point where you could actually sequence DNA and read those letters at such a high fidelity. And it's only been in the last few years that we can do that. So what about these families that have hereditary BRCA1 or BRCA mutations? What does that mean for them? Well, it means they have a very high risk, particularly breast and ovarian cancer. In BRCA1, about a 65% risk of developing breast cancer, and nearly as high a risk of developing a second breast cancer, and a nearly 40% risk of developing ovarian cancer. So we have to think very seriously about what things we can do to screen and prevent these. BRCA2 is somewhat similar numbers, not quite as elevated, but still vastly elevated over the general population. 45 or 50% risk of breast cancer, and 11% risk of ovarian cancer, a little bit lower. And this is a situation where this male breast cancer seems to happen. Well, what are the potential benefits and risks of doing testing in these families? Very similar. We hope we can identify high-risk individuals, and we can also identify those members in the family that did not inherit the mutation and are not at high risk. And it allows us to employ certain early detection and prevention strategies. And here we have actually a number more than we do in the last situation I told you about. The risks are getting lower. We do detect most of the mutations now, but we don't have perfect interventions. We certainly can't screen and prevent all the cancers in this situation. So knowing this information doesn't absolutely mean we can prevent it. So how do we manage these families if a woman finds out she has one of these mutations? Well, there's a number of things that we can think about, and they go from the most drastic and the least desirable, but the most effective to the better. So a prophylactic mastectomy or a preventative surgery to remove the breast cancer tissue at risk of getting a cancer is very effective. It nearly completely reduces the risk of developing a cancer. This is, of course, a drastic and completely unwanted procedure, and the majority of our patients don't pursue this, but some do because they simply don't want to deal with this risk at all and would rather just put it behind them. Surveillance, which means screening tests like mammograms, but more sophisticated ones like MRIs that I'll talk about in a minute, then is our tool to try to pick up a cancer early. And in these families, of course, we're more suspicious. We start at an earlier age. I'll tell you in a minute about tamoxifen, which is a potential chemoprevention agent in this setting, in that it tries to block the estrogen effect on cancers. And then it turns out that a preventative oophorectomy, which is removing both ovaries, reduces the risk of breast cancer by about half. And that's because if it's done in a premenopausal woman, it reduces the estrogen exposure, and that really reduces breast cancer a lot. Of course, it has consequences too, not only on fertility, but on side effects. So this is a tough decision as well, though we often recommend this in women who are completed childbearing, are perhaps 40 or older, are still premenopausal. It can have a significant impact on their breast cancer risk. They have this high risk of ovarian cancer too, and so of course removing the ovaries is very good prevention at preventing ovarian cancer, and so that's why combined this is often a good recommendation. Surveillance or trying to look for ovarian cancer is much harder than breast cancer. We don't have tools like mammograms and MRIs, but there's a number of new diagnostics that are being developed, so hopefully we'll have a better chance at picking up the ovarian cancers. Well, mammography, it turns out, is unfortunately less good in this population than it is in the general population at picking up an early breast cancer. And that's because these women are much younger than the women we're normally screening with mammograms, and mammography just isn't as good there's more density, it's harder to read the mammograms in that population, and there's certain things associated with the kinds of cancers that occur that make mammogram less good. So we have really needed a better tool for screening. And the tool that's emerged over the past few years that's much improved our ability to pick up cancers early in this setting is breast magnetic resonance imaging, or MRI. So MRI, as you know, is a big screening machine, much like a CAT scanner, but doesn't use radiation. It uses a magnetic field to produce an image. And it's been studied in many, many patients now who have BRCA1 or 2 mutations or are at very high risk. And the sensitivity of picking up a cancer is much, much better. And when cancers are picked up in that setting, they tend to be much earlier. We can spot very small ones. And I'll show you in a minute, we can often pick up things that simply aren't seen on mammograms. So we have a clinical trial here that's using MRI in women with BRCA1 and 2, and this is becoming nearly the standard of care for women with a genetic mutation, and is often covered by insurance now. But these are very expensive tests, and so this has not yet gotten extended to a more general population. This is just an example of what this looks like. I don't know if this projects very well here at all. I can't see it. But this is just a standard mammogram, and it turned out that that was totally normal. We didn't see any abnormalities in this woman who had a BRCA1 mutation. But when she had an MRI, and again, I apologize, we just can't see this at all, there was a spot, a very tiny spot that got picked up on the MRI that we did not see in the mammogram at all, and it turned out this was a very, very early breast cancer. So this is the advantage of MRI in this population. It's picking up these early breast cancers when they're hopefully a much more curable stage. Well, what about tamoxifen? As you know, tamoxifen is used commonly in women who've developed breast cancer that expresses the estrogen receptor, or is estrogen responsive. And tamoxifen is an anti-estrogen that helps treat those kinds of cancers. So it makes sense to think, could tamoxifen potentially prevent those cancers from ever arising? And large studies have been done and shows that yes, in fact, tamoxifen can decrease the risk of developing breast cancer, particularly in women that have some elevated risk, some family history. In women who have BRCA1 or 2 mutations, it's not yet clear that tamoxifen is helpful. And that's for a number of reasons. One, because there's far fewer of those women, it's harder to study that, so we just don't have very good information yet. But the second is that the cancers that arise in that setting tend not to have the estrogen receptor, so they're not responsive to blocking estrogen. So it's not clear yet if tamoxifen really is the right agent for prevention. And I'll tell you about a trial we have looking at another agent. All right, so what's going on in the world? How are we getting better at all these things? I told you about a lot of areas that we're not close to perfect or where we want to be in these families. Well, lots of work has been done many, many places to try to get better techniques for screening breast cancer using MRI and some other techniques. I'll show you in a minute some research we have here using a procedure called ductal lavage to try to improve picking up early cancers. Lots of work is going on to try to have better early detection techniques, not just using radiology type scans, but maybe finding a blood test that's very, very sensitive to an early cancer before it can even be seen on a mammogram. We're not there yet, but there's lots of technologies to try to look at much, much larger numbers of proteins, thousands of proteins, and identify something like that. And then prevention, as I said, people are looking at various other agents, other anti-estrogen agents like tamoxifen, other agents that we use to treat breast cancer. And I'll tell you about some work that we're doing here that looks at statins. So drugs that people actually use to lower their cholesterol turns out to have an unexpected effect of potentially preventing breast cancer. And so we're starting to look at that. So let me just tell you quickly about this ductal lavage procedure. It looks and it sounds horrendous, but in fact it's not as bad as it looks. What this involves is actually placing a catheter into the duct of the breast. And that one allows one to collect some of the epithelial cells, the actual cells from the ducts of the breast, and that's where breast cancer actually arises from. And that means that then we can look at those cells, the pathologist can look at those very carefully and see if there's very, very early changes that are associated with early cancer far before you'd ever see a mass on a MRI or a mammogram. And so we're hoping that finding these early atypical changes will help identify women who seem to have some early risk that we should perhaps be watching much, much more closely. It also allows us to use some of the really new, incredible tools from molecular medicine and from genetics to look at very few number of cells for very specific changes. This is just actually a picture of the cells that we can collect from the ducts that we can now actually culture in the lab. You can see they actually start to form little ductal structures. And that means we can do much more sophisticated tests. We can gather the DNA from these cells and we can start to look for very subtle changes in other genes that may be affecting risk of progression. I told you many cancers have multiple genetic changes. We can start looking for some of these other changes. We can actually look at how well these cells function in certain things, and you can't see this, but we've developed ways to examine how well these epithelial cells repair damage that occurs to their DNA, and potentially thus we can predict how much risk a particular woman is for progression. We've combined some of these techniques to try to actually be able to get much, much better images of the breast. Maybe you can see it a little bit there. So this is an image, this is an MRI image of a breast in which we've placed the lavage catheter in a duct and actually instilled some dye. It's called gadolinium, which lights up on the MRI in a different angle. And if you see this at better resolution, what this light shows is this beautiful picture of the branching tree of the duct. So the idea here is to get a very, very specific view of those ducts where these breast cancers start. We might be able to identify small lesions that we can't even see on a normal MRI or some defect that starts very early on. This is very new, and we've only done a few of these, but this is work that's going on here at Stanford. And finally, one of the major goals in this is women who unfortunately do develop cancer, in which we know they had one of these mutations, can we tailor the treatment for that cancer in some specific way? And this is a common theme in all of cancers now. Can we use genetic information to try to pick out from all breast cancers, should we treat this group in a certain way and use other drugs in another way? As you know, to this point, it's very empiric how we do this. It's sort of a guessing game in terms of who we treat in which way. There are certain characteristics about the BRCA gene that makes one think that those cancer cells might be more susceptible to certain kinds of drugs. This is just an example of an experiment from my laboratory in which we made a mouse in which the mammary cells or the breast cells of that mouse, we could either have wild type for the BRCA1 gene, normal, or we could knock out or mutate the BRCA gene. So we can compare these very closely. And here we're looking at the sensitivity to cisplatin, a chemotherapy drug that we rarely think of using in breast cancer. And it turns out that these cells that were missing the BRCA gene are much more sensitive. This is how many of these cells are being killed at increasing doses of this drug. So this would suggest that perhaps we can find certain characteristics of these tumors that make them more susceptible to certain treatments than the 90% of cancers in which this isn't the case. And now we can start trying to use this information to really tailor treatment to certain individuals. Other work that's going on, as I told you, is that we have prevention trials here using statins to try to see if that works as a chemopreventative agent in breast cancer. And there's certain markers that we can use that we can try to tell in an earlier time frame if it's helping. One of the problems with prevention studies is it would take years and years and decades to actually find out if you're preventing cancer in a population. So we want to use some short-term biomarkers to see if we're having an effect. And we have a number of studies in populations trying to find out what are the risks of cancer in other populations and do ethnic differences affect risk, and the cost effectiveness of some of these procedures to try to help justify getting them used in certain situations. Okay, well, I want to finish then by just talking about the future, and this slide is just to remind me that it's all about genetics. So if we look at this slide, two incredibly different people here. But if one actually looks at the DNA from these people, they only differ by 0.1%. So we all share an incredible amount of similarity in all of our DNA, and there's a very, very tiny amount of variability between us that determines not only that we are all different from each other, but also determines health risks, response to drugs, risks for cancer, risks for other diseases. And it is only very, very recently that we can actually exploit that information in a real way. And that's because the human genome has been sequenced. So there's 3 billion base pairs, or 3 billion letters in the genome. So even though we're all 99.9% identical at this base pair level, if one makes the calculation, that means that there's 3 million differences, which is a huge number compared to when you think about 0.1%. Everything I've been telling you about tonight is looking at single genes, looking at 3 or 4 genes that we understood. But we know between any two people there's going to be millions of differences in many, many other genes and many other parts of the DNA. And so we're at a point now where we can actually at one time look at thousands and thousands of different changes and be much more sophisticated and powerful in how we can use that genetic information. And so this is what we now call genomics, which is simply a term that means studying genetics, but just at a very large level, looking at genetics of all the genes at once, looking at all 23,000 genes or proteins. And this technology is here. Incredible advances have been made in the last 10 years, and at some point it's just really expense. And very recently the expense of doing this is getting to the point where in the next 5 years these will really be clinical tests that are going to be start to get used in certain situations. And so I think really in the next 5 to 10 years we are going to understand the genetic basis of many more human diseases, particularly common diseases like cancer, diabetes, high cholesterol. And we'll have a much, much cheaper way to test these patients with genetic tests like I'm talking about, easily tenfold less than the current costs are. And the idea is that we can really use these tests to diagnose disease better and actually understand how to treat patients better. Well, in the cancer realm at Stanford, we have a genomics group. So we have a group of investigators that specifically is trying to apply these new technologies in genomics, looking at high throughput, high scale changes in genes to clinical cancers and using these tests as part of our clinical trials so we can really learn as we go forward which tools we can use to predict who will get cancer and how best to treat it. And so our goals then are to translate these discoveries into the clinic and identify better ways to identify cancer risk and progression, to identify better targets to prevent cancer or treat it, and to better understand how subtle changes in these thousands to millions of letters in the genome affect risk of cancer and other diseases in certain ways. And finally, one of the advances using these genomic technologies that is really here is also studying the tumor itself at this level. And you've heard about molecular profiling, which is a way to look at the expression of thousands and thousands, if not all of the genes from a tumor at one time in order to try to help better predict why that tumor in this person might be different than the same type of tumor in another person because of changes in gene expression. At a very simple level, we, for instance, use this in breast cancer already. You measure whether breast cancer has estrogen receptor or it doesn't, or whether it has HER2/neu or it doesn't. But if you could do that at 10,000 times the level, you might have a lot more power and be able to predict. And so the idea is if you can take RNA, which is the product of the genes from a tumor cell, and look at the pattern of expression of all the genes, or many, many thousands of genes, we might be able to, out of that person, predict who has a worse prognosis, who has a better prognosis, who do we really want to think more about doing more therapy, chemotherapy or radiation therapy, who may not need that at all, instead of treating all of these people just the same, which is basically what we do now because we can't separate those two groups as well. So with that, let me finish and see if you have any questions or other things you want to talk about. So 2 good and complicated questions. The first one was going back to this idea of 50% risk. So again, doesn't mean that the risk of that individual is 50%. It means that there's a 50-50 chance that each child could inherit that altered gene and thus develop all the risks that I told you about in the various syndromes. There's a 50% risk that they wouldn't inherit that gene from one parent, and so they would inherit no increased risk. So it's an all or nothing thing at the level of the chromosomes, of the DNA. Now the second question, what about a situation where we see lots of cancers on both sides of the family? And in fact, we do. And that's where this gets extremely complicated in trying to figure out these things. And so there's no simple answer to that. If both sides of the family seem to have very similar syndromes that might be due to these same genetic changes, that may compound the risk very much. if they're due to different things, that may not necessarily compound the risk. So it's very personalized in these sorts of situations. You really have to look at each family carefully and figuring these things out. Her question was, her son had lung cancer, unfortunately, and should there be testing? Here again, lung cancer isn't one where we've figured out what the genetics are. In fact, the majority, he's so young, this probably isn't the case. The majority of lung cancer obviously is related to smoking and not necessarily to genetics, but that's not the case in this situation. So it's extremely rare that this happens, and this is one of those situations where it's very hard to study because that's going to be a very few people that ever get it at that level. So again, we don't have a clinical test yet to know how to test his kids for their risk. The question is, well, he's asking about a particular lab procedure that tries to predict the sensitivity of any given cancer to a whole variety of different drugs. Again, getting at this exact point at the end about can you tailor the treatment much more specifically. So people have been, for about 20 years now, working on this. It's akin to in microbiology, how you culture bacteria. You can see if it's sensitive to tetracycline versus penicillin. That works great in bacteria. It's not quite as simple in cancers. It seems to be pretty good at telling us what things a certain tumor's not responsive to. It hasn't been quite so good at telling us, oh, here's the things that it is responsive to. But those techniques are getting better, and that's exactly the approach that, combined with some of the more sophisticated things, I think are going to get at that. Can we either using culturing the tumor in a dish or looking at the DNA and other things, or all these things together, pick out what would be the best treatment for that individual as opposed to just everybody that has colon cancer. I told you that for both the syndromes and for all these cancer syndromes, there's sort of a gradient of risk, right? Over age, there might be an 80% chance in a lifetime of getting a cancer, but that goes up and up and up. Some people are getting a cancer when they're 20. Some people with the very same mutation and the very same gene are getting cancer when they're 80, and some aren't getting cancer at all. So what's that about? Well, it most likely has to do with the thousands of other genes that all have very, very subtle effects. So it's trying to understand how does one modify the risk. So you have this one major gene that causes risk, but that risk is getting modified undoubtedly by thousands of other things. The environment, for one thing, but also other genes that you're inheriting. So that's a much more complex question, because now instead of looking at one gene, you need to look at many, many. So this is exactly what people are doing, is trying to understand all these other 3 million changes that occur, that is variation in our genome. Can we pick up patterns there that would help us predict out of all these people that have the same mutation, the same gene, why is some person getting it earlier and one person getting it later or not at all? So the question is how and when and how much should MRI be used in certain situations. So MRI is available at major cancer centers, so it's certainly available here and more at lots of hospitals and not only major places. Its use for breast cancer screening has really been optimized at larger hospitals, and Stanford is one of the, Stanford and UCSF are two hospitals that have done a lot of this, and their radiologists are very expert at screening. It's clear that it's very effective and much more effective than mammogram in women who have BRCA1 and 2 mutations. So that's the particular population where it clearly is effective. Obviously, the next question is, well, should we be using this, instead of mammogram for everybody? Well, that gets to be a complicated question, both for medical reasons and for economic reasons. At this point, MRIs are about $3,000 a piece, and a mammogram is less than $100. It just is not going to be a realistic screening tool for the entire population at this point. It's also not clear that it is a lot better than mammogram for women who are not at elevated risk or are at average risk, who are older, because it's the very young women for whom mammogram is definitely not as effective, and that, of course, in particular affects these BRCA1 and 2 carriers. So if we know what the exact mutation is in a family, so we know what we're looking for. So as we talked about before, there's a 50-50 chance that defective piece of DNA got passed off to any children. If that child inherited it, they now have 2 genes, one of which is defective, and they will have a 50-50 chance of passing it on. But if another child did not inherit it, got a normal copy from mom, a normal copy from dad, it can't get passed on. It's not part of his DNA. So it's not going to come up from no place. So that's another really important thing here in identifying the families, because whole wings of families may not be, this may not be relevant for if it couldn't have gotten passed on there.